So let's talk about the transcription first. Transcription is the process by which a gene in the DNA makes a copy in the form of mRNA. So here is the DNA in the nucleus. A small fragment of nucleus is called a gene, which codes for a specific character in the organism. This gene makes a copy of mRNA on itself in the process called transcription. mRNA carries this genetic information from the DNA outside of the nucleus to the ribosomes, where the protein synthesis occurs. But the process of synthesis of proteins from the mRNA is called translation, about which we will discuss later. So we know that DNA has two strands. One strand is called the coding strand. That is the strand which we read when we have to find what the gene is. The other strand is the template strand, which acts as a template for the formation of mRNA. Just like DNA polymerase performs the formation of DNA strands, RNA polymerase leads to the formation of polymerization of nucleotides to form RNA strand. The RNA strand is also built in the 5' to 3' direction. There are three main steps or stages of transcription: initiation, elongation, and termination, just like you saw these steps in DNA replication. The key features of initiation of replication are: first is the presence of a promoter. Promoter is a DNA sequence where the RNA polymerase binds so that it can start building the RNA strand. Commonly, this sequence of DNA is called the Tata box, where the RNA polymerase comes and starts the replication. But before RNA polymerase can come, there has to be the presence of some transcription factors which help in the binding of RNA polymerase. So these are the transcription factors. Once they have settled on the Tata box, an RNA polymerase will come and sit for making the polymer of RNA. The RNA polymerase also performs the function of unwinding the DNA strands so that an RNA can be built on top of this. Now this whole complex where you have your polymerase, your promoter site, your transcription factors, and a little bit of opening of DNA strand is called the transcription bubble. Next step is the elongation of mRNA transcript. Elongation requires RNA polymerase. The RNA polymerase moves along the DNA and keeps bringing complementary nucleotides, and the synthesis of mRNA occurs. So what does complementary mean? That means wherever there is T, you will have A. Wherever you have G, you will have C in your RNA, and wherever you have C, you will have G in your RNA. But very important point to note is wherever you will have an A on your RNA, you will have U instead of T. Once the elongation has completed, now the transcript has to terminate. The transcription terminates when RNA polymerase encounters a specific sequence of DNA, which is the termination sequence. This termination sequence is usually polyadenylated. Sometimes the sequence is AAU, AAA. When the transcript encounters such kind of sequence, the transcript cleaves off from the DNA fragment and mRNA is detached. In eukaryotes mainly, sometimes post-transcriptional modifications occur. That means the mRNA, which is the pre-mRNA, is sometimes modified. How? So there are some unimportant sequences in the mRNA fragments. These are called the introns. The first process will be to remove these introns in the process called splicing. When you're only left with the important parts called the exons, further modification occurs. One of the modifications is the addition of 5' cap. So you will have a guanine with some protective action put on the 5' end of the mRNA so that the mRNA is not degraded, because it's very important to preserve the mRNA so protein synthesis can occur in translation. Further, there is addition of a poly A tail at the end, which is also involved in stabilizing this fragment of mRNA. The main difference between the transcription in prokaryotes and eukaryotes is the prokaryotes do not have a well-defined nucleus. That's why the transcription is occurring right in the cytoplasm. Therefore, the translation can almost begin simultaneously as the mRNA strand is building. But that cannot happen in eukaryotes because the mRNA has to first exit the nucleus and enter into the cytoplasm where you have your ribosomes, and that is the site where translation occurs. Transcription factors are the proteins that control the rate of transcription by binding to specific DNA sequences. Now, more the transcription factors for certain genes are there will be more copies of that mRNA produced. That means the more protein from that gene will be synthesized. Enhancers or silencers are the DNA regions which can increase or decrease the transcription level. So enhancer would increase the synthesis of mRNA strand or increase the process of transcription, and silencers will reduce it. Thus, transcription factors and the presence of these silencers or enhancers regulates the expression of gene. They can either make a lot of protein from that gene or reduce the amount of protein formed from that gene because the gene is no more expressing. Now, mRNA has copied the information from DNA and it has brought that information from nucleus to the cytoplasm. How will this mRNA be read then? The mRNA is a long chain of nucleotides. In the beginning, it should have one codon. Codon is the set of 3 nucleotides. So in the beginning, it should have a set of codons which are the start codon. In the end, it has the stop codon. The stop codon is UAA, UHE or UGA, and the start codon is AUG. In between, there is information for all the amino acids that have to be put together to form a polypeptide chain. 3 nucleotides are read as one codon, and one codon gives the information for one amino acid. Just like the errors in DNA replication, the errors in mRNA transcript formation can also lead to various problems because this information has to be used for the synthesis of polypeptide chains, which in turn fold up to become functional proteins. Now, if the sequence of amino acids is not correct, you will not have the correct proteins. This leads to various genetic defects, loss of various important proteins, or gain of harmful proteins, which in turn leads to cancer. Now let's talk about the translation process. So the process of forming mRNA on DNA is transcription, formation of protein from the information on mRNA with the help of ribosomes is called translation. So the central dogma says from DNA you make RNA, from RNA you make proteins. So translation is the process in which mRNA sequence is decoded for the synthesis of polypeptide chain which forms the proteins. Translation occurs in the cytoplasm where you have ribosomes. So the polypeptide chain is built on ribosomes. That's why the ribosomes are also called the protein factories of your cell. So the key components that you need for translation are mRNA, on which has to be read, which provides you information about what amino acids to put together. tRNA carries different amino acids and brings it to the mRNA strand so that they can be joined together to form the polypeptide chain. Then you need ribosome, which put all the machinery together. It holds the mRNA on one side or tRNA on one side, and this leads to the polymerization of amino acid chain. Then you need amino acids, which are the building blocks of proteins, and there are several essential enzymes that you need for this process. Now we have talked about how do you read the mRNA information. The mRNA information is the genetic code which is written in the form of codons. So codons are the set of 3 nucleotides. These 3 nucleotides encode for specific amino acid. But how do you read this code? So the code is supposed to have 3 nucleotides. The nucleotides can be A, U, G, C. First you read the first letter of the code, then the second, and finally the third nucleotide of a code. It gives you information of what the amino acid would be. Let's say you have A, C, G. So A, C, G will give you threonine. AUG is the start codon which codes for A, U, G, methionine. And stop codons do not code for any amino acid. These stop codons are where your translation will stop, and these codons are UAA, UAG, UGA. You have to remember these codes. Now let's see again how these codes are read. If you have ATG in your DNA strand, the complementary strand or the template strand will be TAC, because T is complementary to A is complementary to T, and C is complementary to G. When you make mRNA on this template strand, you will have A, U, G. And as you keep on going, you have G for C, C for G, U for A, and G for C, G for C, and U for A. Now let's find these mRNA codons in the chart on your right. So AUG will be methionine. GCU will be alanine. And then GGU will be glycine. Then AUU will be isoleucine, and UGC will be cysteine, and you end up with MAGIC sequence of amino acids. Now, just like transcription, there are 3 stages of translation. initiation, elongation, and termination, because now you are trying to build a polypeptide chain. At the start of translation, first a small ribosomal subunit will come and sit on the mRNA strand. Then the tRNA carrying methionine has to look for the AUG, which is the start codon on mRNA. Once this tRNA finds AUG, the start codon, it sits on it, and the larger subunit of ribosome comes and make a complex with the smaller subunit of ribosome. This initiates the translation process. Now let's look at the steps of elongation of polypeptide chain. On your ribosome, you have two main sites, the A site and the P site. The A site is where the next tRNA comes and brings the amino acid. P site is the site that holds the tRNA with the growing polypeptide chain, and the polypeptide chain keeps building here. The E site is another site on the ribosome where your tRNA, after transferring its amino acid, exits. So the polypeptide chain, as it keeps building, here the tRNA keeps exiting from the E site. Now between the amino acid sequences, there has to be formation of polypeptide bonds, which leads to the formation of long strand of polypeptides. Gradually, the ribosome keeps shifting its position on the mRNA strand to next codon, and the amino acids are brought in by tRNA, and the polypeptide chain keeps building in the P site. Eventually, the ribosome comes across a codon which encodes for the stop signal. This stop signal codons would be, as mentioned previously, be UAA, UAG, or UGA. Once this site is encountered, there is no tRNA for this codon. Now, instead of tRNA, some release factors come and sit at the stop codon, which triggers the separation of ribosome subunits and the polypeptide chain. This polypeptide chain is then ready for folding and for undergoing post-translational modification, which then leads to the formation of properly folded proteins. Let's look at the role of tRNA in translation. So tRNA has the anti-codon site, which recognizes specific codons of mRNA. It also has amino acids attached to the other side, and specific amino acid has specific anticodon which recognizes specific codon of the mRNA. The Wobble hypothesis says that the first two nucleotides of a codon-anticodon pairing are more important. The third one can have a Wobble pairing, which is why you would see in the chart for amino acids that amino acids are more specifically related to the first two codons. Third may or may not be exactly same, so it can vary, and that's why you have more than one codon or anticodon recognizing one specific amino acid. Now, as mentioned previously, the polypeptide chain formed on the ribosome has to then make a properly folded protein. So there are several steps of making protein from a polypeptide chain. The polypeptide chain first has to fold properly to make a protein with specific active sites. If the protein is not folded properly, you will not have proper active sites for the protein to be active or functional. Other than that, several post-translational modification occurs. That means on the polypeptide chain, you add several other molecules such as sugar molecules or lipid molecules. These are called the post-translational modifications. Once all the post-translational modifications have occurred, now the protein should be transported to its site of action. They can either go into the peroxisome, they can either stay in the cytosol, they can go back into the nucleus, they can either be transported into the endoplasmic reticulum or go to the mitochondria. If they enter the endoplasmic reticulum, they undergo post-translational modifications here and can sometimes be excreted or secreted out of the cell. Now, the main difference between the translation in prokaryotes and eukaryotes is because the transcription was not occurring in a specific nucleus in the prokaryotes, the translation could start almost simultaneously with the transcription process in the prokaryotes. But in eukaryotes, there is a specific location for transcription and translation. Transcription occurs inside the nucleus and translation occurs in the cytoplasm. The goal of transcription and translation is to decode the information in DNA, which is the genetic material, and form proteins. But there are many things that can go wrong. Let's have a quick look at that. Maybe you do not synthesize the correct mRNA from the DNA strand. Maybe you are not splicing out the useless information from the mRNA strand properly. At the time of translation, there are possibilities that you do not bring the correct amino acid to the polypeptide chain. The polypeptide chain elongation is terminated prematurely, or there is a shift in how you are reading the code, and that shift in one nucleotide will lead to the whole incorrect sequence of amino acids that you bring in. At the time of protein folding, if the protein do not fold properly, if there are no proper post-translational modifications, you do not bring the correct sugar, or there are some proteolytic enzymes which cleave this polypeptide chain, all of these will lead to the formation of defective proteins or unsuccessful attempt at synthesizing proper proteins. That's why this whole process has to be tightly regulated and has to be checked at different points so that there is an assurance that the process is not only progressing properly, but in the end a correct, properly folded protein will be formed with proper post-translational modifications. Let's summarize the transcription and translation process. Transcription is the formation of mRNA on DNA. It happens in the nucleus. RNA polymerase reads the DNA and leads to the formation of mRNA strand. This ends with the completion of mRNA strand formation. Translation is the formation of proteins from mRNA. This step occurs on the ribosome in the cytoplasm. tRNA brings in specific amino acids which join to form the polypeptide chain. These amino acids are based on the codons of mRNA. Termination occurs when there is an encounter of stop codon, which we know already what those stop codon sequences are. Both these processes together lead to the central dogma, which is the formation of proteins from the information present in the genetic material called DNA.